Welcome to the fourth video in the Nanolive technology mini-series, exploring Nanolive's unique approach to label-free live cell imaging. If you have any questions during the video, please send them to us via the chat box or comments, and we will reply as soon as possible. High-content imaging has already been widely adopted in the pharmaceutical industry and was named as a high-impact innovation by a recent review of pharmaceutical companies. Capturing the responses of multiple pathways at once by observing different organelles simultaneously gives more insights into drug mechanism of action or side effects. Scientists are taking advantage of the rich data from high-content imaging to help them better understand cells' phenotypic responses to therapeutics earlier in the drug development cycle, and to improve the translatability of new drugs to clinic. So what does high-content imaging mean to us at Nanolive? High-resolution images to allow us to measure fine details. Label-free, so that we are measuring unbiased responses without adding chemical dyes or fluorescence exposure. Dynamic data to capture rates of activity and time-dependent events, and automated analysis to extract the high-content information captured for more informed drug development. Nanolive's high-resolution imaging allows the investigation of cells from the population level, quantifying confluency or count, down to the single cell level to understand morphology and phenotype, and also to the organelle level. The resolution is excellent and allows for the study of organelle dynamics like the mitochondria you see here. Fluorescence is commonly used in high-content imaging, but Nanolive's unique approach to holotomography makes it possible to do so completely label-free. Let's explore why label-free imaging is so important for the phenotypic study of cells, then cover some of the insights that can be gained with Nanolive's automated analysis for high-content imaging. The ability to gain high-content insights into cellular processes without using harmful chemical markers or fluorescent light is a core principle of Nanolive's holotomographic imaging. As well as allowing long-term investigation of dynamic cell processes, label-free imaging enables the capture of more biologically relevant responses from cells in an environment closer to their in vivo state. This is especially important for sensitive cells like primary cells or iPSCs that can be easily damaged by probes and markers. Label-free imaging is a key tool for studying drug safety and toxicology without probes biasing results. A label-free approach can improve data reliability, therefore improving the translatability of drug development data to the clinic. If a picture is worth 1,000 words, live cell imaging is worth 1,000 endpoints. By avoiding phototoxicity and fluorescence, Nanolive systems can image cells frequently for long periods of time in the controlled environment of our top-stage incubator. Sensitive cells can be monitored without interference, preserving these healthy cells for other downstream assays after imaging. Rates of activity like cell death rate or rate of phagocytosis can be quantified and compared. Time-dependent effects like a peak in T-cell killing activity or contact with target cells can also be detected, again without labels. As we just saw, live cell imaging vastly increases the amount of data captured from a single experiment. Fortunately, machine learning analysis can simplify data collection and interpretation of these images, and at Nanolive we have developed easy-to-use tools which do not require any manual segmentation or algorithm training by the user. To explain how our analysis works, first the software detects cell boundaries, defining each individual cell. Then automated measurements are carried out to calculate the area, perimeter, and other morphological features of each cell. This gives us phenotypic metrics, and we can detect how they change over time with our time-lapse imaging, giving powerful insights into how cells respond to changing conditions and treatments. Because we use holotomographic imaging, the whole process is totally label-free. To detect different modes of cell death, a trained AI algorithm uses these measurements to classify cells as apoptotic or necrotic in the live cytotoxicity assay. Unlike methods that rely on fluorescence markers, this label-free method provides confidence that the cellular responses measured are due to the therapeutics tested and not a side effect of phototoxicity. Another label-free analysis solution unique to Nanolive systems is the live T-cell assay, which can be used to quantify how drugs or T-cell therapies perform against their targets in vitro. After cell boundary detection, this digital assay classifies cells into two subpopulations as either adherent target cells or effector cells. It is the only label-free solution for quantifying the dynamic T-cell response at high resolution and can provide information on T-cell target cell contacts and killing dynamics. Finally, the smart lipid droplet assay works on the same principle of cell boundary detection, then uses the refractive index to detect and quantify lipid droplets within cells. Lipid droplets are connected to many metabolic processes, from metabolic disease to aging. The assay can also be extended to drug perturbation studies, including the assessment of phospholipidosis. Although Nanolive imaging systems work label-free, fluorescence can also be used and quantified as an optional step to gain functional information and to divide cells in co-culture into subpopulations. Using a single marker, the morphological changes and death rate can be measured separately for two subpopulations in co-culture, opening the door for assessment of drug selectivity in more physiologically relevant in vitro models. These different assays can be used to analyze the same acquisition and can even be carried out retroactively, providing deep phenotypic insights to better understand cellular response. And our team continues to innovate to expand the abilities of our high-content imaging. We want to go further to quantify changes in more organelles and their interactions with each other to reveal even deeper insights into cellular responses. This video shows Nanolive's latest research in collaboration with the Institut Pasteur, where we developed a dedicated pipeline for the digital segmentation of mitochondria, nuclei, and nucleoli in cells infected with COVID-19. For examples of our high-content imaging in action for drug discovery, or applications ranging from immuno-oncology to metabolism and many more, please see our past webinars or read some of the application notes on our website. This has been the fourth video in our mini-series. To explore the content of all 4 videos in more detail, visit our website. There you will find more content and a summary of the information in this mini-series. Our team will be happy to answer your questions and provide you with any additional information you need. Please get in touch via the contact form link in our description, on our website at nanolive.ch, or by email at lookinginsidelife@nanolive.ch. Thank you for watching.